All right. Good morning, everyone. I'm Jerry. I'm a rising junior, and I've been working in the Snyder Lab at Stanford this summer. And yeah, so today I'm going to present about my new end-to-end single-cell proteomics workflow. So it goes from just raw spectrometry spectra to more precisely annotated cell type clusters. And yeah, so here's a basic outline of this presentation. So first, I'll go into the motivation. So, why single cell proteomics? Uh, what is the purpose? And then I'll go into the workflow itself, some key challenges and solutions to those challenges that the workflow provides, and some results and some integration strategies, and finally, some next steps and takeaways. So, in terms of motivation, this first point is about why proteomics or why single cell in general. So, basically, bulk analyses will mask these rare cell types, right? They're like averages. So by looking at the single cell level, you're more able to uncover these more rare cell types that might be driving disease and development. Uh, as well as why proteomics over mRNA or RNA-seq. Essentially, RNA and proteins aren't super correlated. There's about like a 30% correlation. And so, since proteins are generally to be thought to be the like workhorses of the cell, like the functional pieces, looking at the proteomics directly will probably give us a greater insight into what's actually happening in the cell. And yeah, finally, by looking at this, what is the final point? We can look at different signaling pathways and find different biomarkers for diagnostics. So a little bit of background. Uh, Processing pipelines for bulk proteomics and scRNA-seq are well documented and they've existed for a while. But for single cell proteomics, this is less documented and there's no single like processing pipeline for proteomics or single cell proteomics. So the goal of this project is essentially to establish this for LCMS data. Oh, yeah. So yeah, just to reiterate, just to establish workflow for analyzing SCMS data and essentially integrating it with the scRNA-seq data from similar samples with the objective of identifying cell types. Yeah, so some key challenges associated with this process of creating the pipeline. So first, as with any processing pipeline, this idea of biological versus experimental variation, right? You have to distinguish between, what is a true biological difference and what is just technical noise. And yeah, especially with, these batch effects that can occur, how do we get rid of those? Additionally, these two main modalities, the proteins and the mRNA, right? The mRNA is a lot less stable, a lot less abundant. So how do you account for these differences? And as well as the differing resolutions of the SCMS versus the RNA-seq technology, right? The dynamic range of the RNA-seq is like a few orders of magnitude larger than the LCMS. So how do we account for that? And then finally, like how do we deal with this underlying data? What kind of conclusions can we make based on this? All right, so just a little overview on the pipelines. So for RNA-seq single cell, there's usually on the order of 100,000 cells per experiment compared to single cell mass spec, which is on the order of like 1,000 cells, right? Uh, Usually RNA-seq you can get on the order of 10,000 genes as well, versus like 1,000 genes or like about 10 to the fifth peptides or 10 to the fourth peptides, um, like measured across the entire experiment. And so the major problem with that is this idea of missingness, right? So with RNA-seq, right, the the missingness is mostly random, right? But with the LCMS, the missingness is mostly not at random. So the imputation method there has to be different. Additionally, like the intensity distribution is very different between these two types of acquisition. So we have to adjust the distributions to match. And then in terms of normalization, there are different methods there too. And yeah, batch effects uh, also different. And yeah, so just an overview of the data that I was working with. So transcriptomics was previously published 10x RNA-seq data. It's from polyp cells. It was about 100,000 cells from 100,000 cells with about like 20,000 genes. And the LCMS data is about 2,000 cells with 44,000 peptides with about like 6,000 genes. And so, yeah. So, yeah, this is just an overview of like the LCMS acquisition itself. So, how do we get this data from these single cells? So essentially, you separate the data, the cells into single cells, right? And you lyse those cells, and then you take out uh, peptides from those, or you take out the proteins from those, turn them into peptides, ionize them, essentially just measure the spectra. And yeah, so as you can see on the right, uh, there were 4 batches, or sorry, 8 batches with 4 different plates. And then these batches and plates were of various sizes. So yeah, that would affect the downstream batch correction process. And so yeah, so the basic, uh, like a very high level view of the single cell proteomics pipeline, you begin with the spectra, and then from that you get the peptide intensity matrix from something like DINN, which is just a neural network that matches the spectra to individual peptides and accounts for their intensity. And then from there, you do quality control, batch normalization, and correction. And then you impute missing data potentially. That's maybe optional depending on your downstream task. And then you aggregate these peptides, right, into the proteins that they came from. And then you log transform in order to normalize the data, like make the distribution more normal. And then from that, you have a processed protein intensity matrix that can be used for downstream analysis. So some basic QC, right? Basically, as you can see, there's a like a very skewed distribution of intensity per cell, and there are some outliers with the peptides as well. So essentially, removing those from the data set helps to remove noise. Yeah. So when we view the summed intensities of cells over time as they were acquired, we can see like clearly like the cells within each batch have like a different distribution, different means, different medians. And so uh, what is the underlying cause? It's not likely that the samples were actually biologically different, right? Difference is likely caused by some kind of technical factor. And so we have to account for that, right? So what is a batch effect, right? It's a systematic technical non-biological variation that's caused by processing in separate groups, right? And so this can be caused by a variety of factors, uh, mainly by like the handling of the different samples, right? There's no way to guarantee that's identical. And yeah, we can see that there are 2 types of variation here, right? Or batch effects here. One is by plate, and the other one is by the sort batch or the MS batch that they were run with. So our key assumptions before doing this batch correction is that the data is missing not at random, right? So if something's missing, it's likely due to low abundance and not some random dropout. And this is true because it's a data independent acquisition. And yeah, we also assume that the correlation between unrelated peptides should be close to zero. And then the correlation between peptides from the same protein should be positive, right? Because of the protein's higher in abundance, you would expect all different peptides to also be higher in abundance and vice versa. And so the main batch correction methods that are being used in these single cell pipelines is combat uh, and then the median correction. So combat uses like a naive Bayes sort of model to try to adjust for these batch effects, while the median is a much more simple model that just adjusts the median of both the protein and the sample to adjust these cells. Just visually speaking, seems like the median has done a little bit better of a job of integrating these batches with each other, whereas the combat you can still see these like batches separated out very clearly. And then along with that, we see that combat also seems to increase the correlation between non-related peptides, which is not what we would necessarily want to happen, right? And whereas the median seems to preserve these distributions. Okay, the next step is just to impute the data, right? Now that we have these values corrected, how do we make this turn the sparse data into more dense data that we can perform analyses like PCA or HuBMAP with? So essentially, what you have to do first is to get rid of peptides that are extremely low abundance, so less than 10% presence. And that is just to get rid of essentially noise, right? If you impute based on too few things, you're just adding noise basically. And then there are a few options for imputing. Um, so zero imputation, mid-intensity, KNN, and normal distribution. And each of these imply like a different rationale for why the data is missing. For missing out at random, uh, generally you would probably use KNN or mid intensity. And then because our missingness is so high, mid intensity is the choice here. And so, yeah, this is the final recommended pipeline. So you go from the peptide intensity matrix, you do this feature level QC. So you remove the PSMs, so the readings with high false discovery rates. And then you remove protein groups that aren't seen in a lot of samples. And then you sample over QC. So you remove samples that don't have a lot of peptides or proteins. And then you remove samples that have either too much intensity or too little intensity, because too much intensity implies, potentially that you have a doublet, and then too little intensity implies potentially that you might have had like an empty well. And then following this basic QC, you do median batch correction per batch per plate, right? So you get all batches or all cells on a single plate to batch up, and then I do median batch correction per plate. So finally getting all these cells from different plates to match into similar distribution. And then if needed, you do uh, 1/2 sample main imputation. So you take the minimum intensity in any sample, and for all the peptides that it hasn't seen in that reading, you impute the 1/2 sample min. Yep. And you aggregate the sum of the intensities to get back to the protein level, and you log transform that so that it's more comparable with the results from an RNA-seq output.
All right,
And
we
yeah, so
will
this,
ask you to just speed it up a little bit as you get to your ending. We do still have one
okay,
more presentation. Thank you.
sure. Okay, so looking at the RNA-seq HuBMAP in comparison to the uh, just SCMS clustering. And so we can see that the differential expression of genes within clusters in RNA is a lot more, right? It's a lot more differentially expressed than in protein. So yeah, so it's a lot harder to find these markers within these protein modality. And so one strategy to integrate these two cells is for every identified cell type in the RNA-seq data is to just identify the markers and then measure the aggregate expression of these genes in the MS data. But yeah, as you can see, some cells seem to map pretty well to those in the MS acquisition, but other cells are less obvious. And uh, yeah, so this integration strategy might not be the best way to do it. Uh, Another strategy is just to compute the correlation between the top differentially expressed genes, right? So the marker genes from the RNA cell types, and then just find that correlation with the SCMS data. And then we can see that this is also not like a great integration strategy. So right now, there isn't like a great strategy. So we're still working on potentially finding a better way to do this, potentially with like a machine learning model, or something like that. Yeah, so like a key takeaway is that yeah, the key takeaways are essentially the end-to-end workflow reduces noise while like maintaining the biological variation, right? Um, by assuming that missing data is not random, guides our choice of imputation, and then batch corrections do have some trade-offs. Potentially future directions, right? We could make like a web-based version, making it much easier to create these pipelines and validate them. Uh, Machine learning based model to map cell types, do this integration, couple clustering along the same lines as that. And then I can use RNA-seq to potentially predict these missing values instead of just estimating by minimum value. And then you can do a multiple regression analysis to predict like the contribution of, for example, mRNA within like the protein. And finally, yeah, I'd like to thank my advisors, Tianchen and Li Hua, as well as my PI, Michael Snyder, and the HuBMAP consortium as a whole.
Thank you. Um, I just wanted to ask, we'll just do one question, sorry. But what do you think is the most valuable thing you will take away from this entire experience?
I think it's like mostly about like asking the right questions, which directions to go in. Sometimes if you don't think about like what direction to go in first, you can go down like a lot of wrong paths, I think.
Awesome. If anyone else has any other questions for Jerry, please put them in the chat, and we'll get started with Caden so that we can wrap up on time. Thank you.